colleagues, distinguished professors, distinguished representatives, distinguished participants, I welcome you to the award ceremony. Among the oral presentations and poster presentations, the best research awards will find their owners. And in order to present the third place award, I would like to invite Şaziye Senem Başgül to the stage, please. And I would like to announce the third winner. And I tracking
neuropsychological profiles and reading performance of children with attention deficit hyperactivity disorder and dyslexia. Ödülünü almak üzere
in order to receive the
Dombaloğlu'nu sahneye
token of appreciation, I would like to invite Mr. Dombaloğlu to the stage, please. And in order to present the third place award, I would like to invite Ms. Sakalı. And I would like to announce the winner, the oral presentation.
neuroinflammation and hippocampal atrophy in rats with induced post-traumatic stress disorder. Ödülünü almak üzere
And in order to receive the token of appreciation, I would like to invite Dr. Sena Karaca. And for the second place award, in order to present the token of appreciation for the second winner, I would like to invite Mr. Ömer Yamarşa. And I would like to announce the second place award.
Post-weaning social isolation stress leads to behavioral impairments and focus on the alterations of the hippocampus and prefrontal cortex. Ödülünü almak üzere
In order to receive the token of appreciation, I would like to invite Dr. Gülşen Çelebi. And I would like to announce the winner, the
period.
second winner
Efficiency and safety are
of the
demonstrated with
oral presentation.
example targeting the NPFC with a double cocktail in treatment-resistant depression. Ödülünü almak üzere
In
Sayın
order to
Profesör
receive the
Salih
token of
Altunç'u sahneye davet ediyorum.
appreciation, I would like to invite Mr. Altunç.
Bir birincilik ödülüyle
And
devam
for the
ediyoruz.
first place awards,
Birincilik ödülünü vermek
in order
üzere
to present the
sevgili
token of
Ferdi Bey'i sahneye
appreciation, I
davet
would like to
ediyoruz.
invite Mr. Eric Vermitten to the stage, please.
Kazananı
And I
açıklıyorum.
would like to announce the winner.
Birincilik ödülümüzün
The
tamamı
first place
için
award goes
our
to
environmental
our
conditions and access to resources related to children's brain and cognitive development. Ödülünü almak üzere
to
Sayın
receive
Yaman Yıldız Özhan'ı sahneye
to
davet
receive the
ediyorum.
token of appreciation. I would like to invite Neslihan Özyıldız.
Sunumunu yapmak üzere
And
Sayın
for the
Sena
presentation, I
Karaca'yı
would like
davet
to
ediyorum.
invite Ms. Sena Karaca to the stage, please. Hello to everyone. My name is Sena Karaca, and I would like to talk about the effects of ketogenic diet on neuroinflammation and hippocampal autophagy in rats with included post-traumatic stress disorder. And I would like to present you in short, so I would like to talk about the methods. In our study, we had 24 adult male Wistar rats, and these were divided into 3 groups, and PTS model was not induced in 2 groups using the single prolonged stress protocol, and this was the PTSD. The model was used as a single prolonged stress protocol was used, and for 2 hours, psychological stress was created and squeezed them, and then afterwards, for 15 minutes, we had them swim, and then for the chemical stress, we exposed them to ether. And then afterwards, 1 month social isolation in order to increase the level of the stress. And then we fed them on standard or ketogenic diet for 28 days, and we assessed the behaviors. And for this test, we use the peaceful room for the elevated plus maze test. And then in the methods, in the meanwhile, with ELISA, TNF alpha, IL-10, and LC3 levels are also measured. And to the best of our knowledge, this study is the first to investigate the effects of a ketogenic diet on post-traumatic stress disorder in an animal model. And it's a preclinical study. And along with this, and the pathophysiology of the PTSD, microglial activation is present, and the ketogenic diet polarizes. And then we have all the findings related to this. And related to autophagy statistically were not found to be statistically significant, and related to this, a ketogenic diet for prevention of PTSD, whether it would be influential or not, whether it has a therapeutic effect or and on which days autophagy is effective or not. In order to identify this, of course, we have more further studies clinically on this. Thank you very much.
Teşekkür ediyoruz.
Thank you.
Sunumunu yapmak üzere
And for the
sevgili doktor
presentation,
Adolescent
I would like
Dombaloğlu'nu
to
sahneye davet ediyorum.
invite Mr. Mehmet Ali Dombaloğlu. And good evening to you all. First, this Congress, for the organization of this Congress, I would like to extend my professors as well as to the participants. I would like to extend my thanks, and also I would like to thank my thesis consultant, for providing support for this thesis. And in our study, the psychiatric disorders, the ADHD and dyslexia, the cognitive functions and also reading performances were compared, and ADHD and dyslexia are among the most common psychiatric disorders in childhood, and both can negatively affect the cognitive functions, and also we can have difficulty in diagnosis. And in terms of functional functions to be we thought that with this study we could get good results out of it, and also we had the dyslexic and also ADHD children were compared with the normal individuals, and then we had the method of evaluation with eye tracking, and I would like to elaborate on the CNSBS, and it includes 10 tests, and it checks 6 behavioral fields, and the keyboard is used here in the test, and there are many tests included, and we have the oral, and then the cognitive flexibility and continual performance tests, and composite memory, complex attention neurocognitive indices. We used the multi-testing context, and then with the eye following. So there is an enlightening light source, and then there is a reflection. The reflection is being followed by the camera. And during the reading, there are two activities: fixation and saccade, and the fixation is where the eyes just fix on certain points, and also we have the saccade that enable the transmission from one place to another. And we had 74 children, and we had in the healthy individuals 26, and 26 in the ADHD, and 24 in the dyslexic children, 26 as well, sorry, and none of the participants were using psychotropics, because the psychotropics has certain effect on the patients, of course. And if we look into the results, and CNSVS, this neuropsychological tests, as expected, neurocognitive index and complex attention and cognitive flexibility, ADHD and dyslexia group perform weak compared to the normal individuals. And in the memory group, the verbal memory was bad in the group of ADHD. As an interference for the clinically, the ADHD group should be provided more visual items that would contribute to their education. And as a result of the eye tracking, in the dyslexia group, compared to the ADHD and the control group, they differentiated. It differentiated, and the group of the ADHD eye movements compared to the control group did not show a significant difference, and the ADHD and the control groups are similar, and dyslexia group differentiated from both of the others. And here in fixation and saccade, we made the evaluations on the words, and we identified each word as an area of interest. And in the control group, healthy control group, the eye trackings were like this, and the ADHD eye movements were like this, and every round thing is a fixation, and in between the saccades, and in dyslexia there were more saccades and fixations. And while we provide psychotherapy or psychoeducation to the parents, it is a bio indicator, especially for the dyslexic children. And in conclusion, our study supports the view that the ADHD and dyslexia cause significant impairment in various cognitive domains, and shows that the eye movement of children have a role in this. And thank you very much for your listening. Thank you.
Teşekkür ediyoruz.
Thank you.
Sunumu yapmak
And
üzere
as to for the presentation, I would like to
Adolescent
invite Dr. Gülşen Çelebi to the stage, please. Distinguished professors and participants, I'm a research resident in pharmacology, and I'm very much, I'm very, very happy to have been awarded for this. And I'm taking this opportunity, I would like to thank to my professors as well as the organizing committee for this. And as the oral presentation, I have taken the doctorate thesis. Post-weaning socialization stress leads to behavior impairments, a focus on the alterations of the hippocampus and prefrontal cortex. And this is the flow of my presentation. And the early life stress has been associated with long-term changes in stress, reactivity, emotional regulation, and cognitive performances. And despite the critical role of adolescence in brain maturation, for example, when the pregnant animals were exposed to stress, and it's continues with the maternal separation, there are models like this. And after the weaning, in animals, we exposed them to isolation. And why did we opt for this? The social stress, isolation stress, has been studied because of the impact of the COVID-19. And however, there are no studies on this, so this makes our study unique. And the COVID-19 has got the increasing effect on isolation. So for that reason, social isolation, the effect of the social isolation effect have been identified into two. First, we looked into the behavior level and obsessive compulsive similar disorders, and also cognitive functions were evaluated. And in the second stage, molecular changes were scrutinized. Inflammation and oxidative stress markers were looked into. And methodology, if I had to summarize it, after the weaning, in the postnatal, on the 21st day, we divided the male rats into two groups. One of them is control group, and the other one isolated, social isolation group. Social isolation group, the experimental animals were accommodated individually and as to see the environment as well as each other. And then we use separators for this. And following the 8 weeks isolation protocol, locomotor activities were evaluated first, and then the possible findings we did this in order to exclude that it is due to the locomotion moments. And then depression similar were evaluated with the obligatory swimming test, and then repetitive actions were evaluated, and as well the longitudinal memory was also evaluated with this test. And then under the anesthesia we sacrificed, and then we collected blood and the brain tissue, and also we had a statistical analysis of the students test, and then locomotor activities. There was no difference. However, in the isolated animals, there has been a significant weight increase. And as per the groups in isolated animals, they performed anxiety similar activities, and depression similar activities, and emotional and spatial memory distortions and repetitive types of actions, activities, and movements were identified. And if we look into the molecular results, we see that BDNF were decreased, but the gene expression level, BDNF gene expression increased significantly, and also TNF alpha and ionos, they increase in the hippocampus, but not in the hippocampus. However, the PPR and in the early time, the exposition to the stress in these experimental animals in the prefrontal cortex and and BDNF protective mechanisms make a show an effort and for the compensation. And despite this, the behavioral distortions and disorders continue. And when we look all our findings, the social isolation stress would cause molecular changes, and with different mechanisms cause this. And prefrontal cortex is more sensitive to social isolation, and for that reason, region-specific treatments, oxidative, especially suppressing the oxidative stress, hyper gamma activation treatment approaches would decrease the isolation stress diminishing reduction. And in my PTR gamma agonist effects, I'm studying on this, and the results are very attractive and really attention drawing. And thank you very much for your attention. And I would like to mention my consultant professor here, as well as I also would like to thank to the organizations who supported me financially as well. Thank you.
Teşekkür ediyoruz.
Thank you.
Şimdi sunumunu
Now,
yapmak üzere
for the
Sayın Doktor
presentation,
Ali Tarık Altunç'u sahneye davet ediyorum.
Dr. Tarık Altunç. Hello. And while I start, I would like to thank to the organizing committee for giving us this opportunity. And of course, in my thesis, my resident colleagues, Yusuf Ahmed and also Bushar Poyraz, my consultant, I would like to extend my thanks to them. And in this study, dorsomedial prefrontal cortex, we TMS that was speeded up by fourfolds, and we tried to find out the reliability and safety. And then this is the first study where such an application has been done. And I would start with a method, and in the psychiatry political Cerrahpaşa University, the patients with resistance to drug, at least resistance to one drug, with the psychiatric problems, and for TMS, the patients with the contraindication, we excluded them, and the application cool DB8 double cone coil, and then in every session, 600 pounds left to left hemisphere and to right hemisphere we gave to the brain. And then in between these sessions we had 15-minute break, and in total, we had 4 sessions a day and 5 treatment days. We evaluated the patients. If there is less than 25% response, we terminated, and if the patient is in remission, then to be terminated, and if it is more than 25%, but if there's no remission, then we added 10 more sessions. And before TMS, on the third day, fifth day, and also for the eighth day, and also in the first month, we evaluated patients. And 34 patients were included into this study. One of the patients dropped out of the study due to the suicidal thoughts during the treatment, and one of the patients had severe headache, and one they left without any reason. And then our group included a moderate level depression, and Hamilton score was 21 as the resistance stage. It is the moderate resistance. They had the moderate resistance, and in 61%, in none of them, no side effects developed. The most frequent one is the 29% headache, and also the by 90% there was the sleeplessness, and there was no life-threatening side effect was observed. And in all scales, there was found to be a significant decrease, especially in Hamilton. The median age of the group was 21, and this is the most important result of the of the study. Sorry, and so 65% of the patients has been a decrease by more than 50% in Hamilton score, and the other one went into remission, and these are the back depression scores and this is the trend of the patients who respond and who do not respond. And in the first month, we made an evaluation again. 3 patients received EKT. They were excluded. And then the 23 patients, 65% of them, the response criteria and also the remission criteria are obtained. And when we look into these data, 2 of the patients, just following the end determination of the treatment, we see there is a recurrence, and then and 2 patients in the 1 month follow-up ensured provided the remission criteria. And the first month remission, Hamilton score found to be 10, and with the termination of the treatment, the change in the Hamilton score was not found to be statistically meaningful, significant. And these patients who respond to the treatment are represented with one line, and this is also about the ones who did not respond. So this study was made or is the first to use an accelerated TMS protocol for 4 sessions per day to target the DMPFC. And then there was also a study where it increased the efficacy. However, it was not found to be significant. And working 4 times a day, this may show that it is significant. And prefrontal cortex and the TMS work seems that it increases the efficacy. And also in terms of tolerability, it seems good. And there is a dropout rate is about 8%. It's original. And there is also 13% dropout, and there are 2 inaccuracies, unclear to how long this treatment would be persistent, and also the response trends. 2 of them rapidly respond, and then 2 of them recurred current rapidly. And there are studies that support these findings. And then in the 1 month follow-up, we see that the response goes down from 90% to 70%. And also one study, while it was 50%, it grew up to 55%. In some of the patients, there is a rapid response, but some of them respond late. And also there is a need to go further other studies, and there is a limitation. One of them is about the small samples, and in all TMS studies, the samples are with this size, and also the open label nature of the work, and placebo control group was missing. So the placebo effects could not be interpreted. So this was a limitation, and thank you very much for your listening. 50 minutes of break. There was 50 minutes of break. Thank you. And finally, for the president, Dr. Neslihan Yıldız Özsan, I would like to invite her to the stage, please. Have a nice evening. First of all, I would like to thank for this valuable award, as well as for all these congresses, as well as this valuable association. I would like to thank to Mr. Mesut, as well as all my professors, and as well for giving me this opportunity. I would like to explain and tell about my project in English.
I'm really honored to receive this award, and I would like to thank the Turkish Association for Psychopharmacology for this great organization and bringing us all together. Today, I would like to briefly share our study on our environmental condition and access to resources, as we refer as neighborhood advantages, can be related to children's brain and cognitive development. My name is Neslihan. I'm a postdoctoral research fellow at Harvard Medical School. I'm working at Psychiatry Neuroimaging Laboratory, and we use cutting-edge neuroimaging techniques to investigate brain alteration in neuropsychiatric disorders. As our study's background, children who grew up in a disadvantaged neighborhood previously showed that, in previous literature, this group of children showed that decreased cognitive function, impaired neurodevelopment, and increased risk of psychiatric disorders. Even though some studies investigated these associations, they're mostly focused on the neighborhood socioeconomic aspects and not comprehensively investigate the neighborhood other domains like education or environmental factors. So we hypothesized that increased neighborhood advantages are associated with enhanced neurocognitive function in children, and educational, socioeconomic, and environmental domains of neighborhood may have a distinct effect of neurocognitive development. Can we click the video, please? Thank you. For investigating neighborhood advantages, we used a comprehensive measure called Child Opportunity Index. This index investigates 29 different neighborhood indicators based on children's residential addresses, zip codes, and it gives us an overall neighborhood quality and advantage score, as well as 3 domain-specific scores, which are education, social, economic, and health and environment. And you can see a couple of examples of these indicators, the neighborhood indicators that we used. This is a summary of our methods. As you can see, we investigated over 6,000 children aged 8 to 10 years old. We used a Child Opportunity Index for neighborhood analysis. For statistical methods, we used a generalized additive models controlled by familial and demographical factors. And in addition to generalized additive model, we also investigated neighborhood effect of cognition, how much of the neighborhood effect of cognition can be explained by brain development using mediation analysis. As our outcome measures, for brain development, we focused on the 20 long-range intrahemispheric association tracts using harmonized quality control diffusion MRI data. And for cognitive analysis, we used an NIH toolbox to investigate different cognitive functions like language development, attention, memory, processing speed, as well as composite neighborhood scores. This is a summary of our results on neighborhood effect of association tracts development. As you can see, after controlling various factors related to neurodevelopment and neighborhood, we've specifically found a significant track which called superior longitudinal fasciculus 1. We found this track significant across all neighborhood domains. So increased neighborhood advantages are associated with more development in this specific track. And this track connects important regions called frontal, parietal, and temporal lobes. This is a component of a dorsolateral component of SLF tract and involved in various cognitive functions like memory, attention, and language. An impairment in this tract previously found associated with ADHD, autism, psychosis, and different psychiatric disorders. So this highlights the importance of development in this tract, especially. And this heat map summarizes association between neighborhood and cognitive functions. As you can see, we found a various association in different cognitive functions. The highest associations are in the language development and composite cognitive developments. And with pediation analysis, we found out that neighborhood advantages association with cognition partially explained by this specific tract development and microstructural properties. As conclusion, our study highlights the important role of neighborhood advantages on neurocognitive development, and neighborhood-level interventions like improving educational, environmental quality, or addressing socioeconomical disparities may foster children's neurocognitive development and reduce psychiatric disorder risks. This is a cross-sectional analysis, and even though Neighborhood Child Opportunity Index is a comprehensive score, it still lacks time-based and household-specific details. In the future, longitudinal data sets combining these factors could be beneficial. Thank you for listening to all. I would like to thank our lab and my mentors, especially Dr. Marek Kubicki, who is here with us. And I would especially thank to Dr. Soheyla Çetin Karayumak. She's a mentor of me. And thank you, everyone. If you have any question, I will be happy to answer.
Saygıdeğer hocalarım ve değerli katılımcılar. Ödül törenimizin sonuna gelmiş bulunmaktayız. Ödül sahiplerini tekrardan tebrik eder, katılımlarınız için teşekkür ederiz.